Let us discuss these questions from the NCERT book. The first question is fill in the blanks question. So, there are some statements given and we have to complete. This is the first question. This is fill in the blanks one. A. First statement is humans reproduce, and they have given two options from which we have to choose. Humans reproduce, the two options are asexually or sexually. So, the answer is, because in human beings we find sexual reproduction, gamete formation and their fusion. B. Humans are. There are three words which are given and we have to pick from those. Oviparous, that is egg laying. Viviparous, that is giving birth to young ones, and ovoviviparous. Human beings, they are viviparous because they give birth to young ones. So, the second blank would be viviparous. C statement is fertilization is dash in humans. Two words, external and internal. External fertilization is when the gametes are released into the outer atmosphere, that is, it could be water, and in water if fusion takes place. But in case of human beings, the sperms are deposited in the body of the female and fertilization takes place in the body of the female. So, it is internal. Fourth one, D, male and female gametes are. Two words, diploid or haploid. They are haploid because gametes are produced as a result of meiosis. So, they are haploid. E. Zygote is diploid or haploid. How is the zygote formed? By the fusion of these haploid gametes. One sperm, other egg. So, the zygote is diploid. E, and then the next statement is the F1. F1 says the process of release of ovum from the mature follicle is known as, when the egg or ovum is released from that Graafian follicle, the process is known as ovulation. Release of ovum is known as ovulation. Next is ovulation is induced by a hormone called, Graafian follicle ruptures when the level of LH, luteinizing hormone, is at the peak. If you remember the complete menstrual cycle, on the 14th day there is a peak of LH which is achieved, and that is responsible for the release of egg. So, ovulation is induced by LH, that is luteinizing hormone. Next one, H1, the fusion of male and female gametes is known as, there are two terms, either it is called fertilization or it can also be termed as syngamy, fusion of two gametes. Let us take the I statement. It is fertilization takes place in. So, now they are asking about the location, position, because in these last statements they have not given us the options to choose from. So, fertilization takes place in the fallopian tube, but where in the fallopian tube? At the place where the ampulla and isthmus, they are connected. So, it is known as ampullary or ampulo-isthmic junction of fallopian tube. So, we are talking of fallopian tube, but we are giving even more precise answer. Where in the fallopian tube? At the junction of ampulla and isthmus. J statement, zygote divides to form which is implanted in uterus. Now when zygote starts to divide, we get 2 cells, the blastomeres, then it divides into 4, 8, 16, 32, which is implanted in the uterus. So this first stage which is formed is called morula, the 32 cell, 16 or 32 cell stage. It comes into the uterus, and then when it comes into the uterus, it starts absorbing that secretion of the uterine endometrium. It develops a cavity. After a cavity is formed, we start calling it blastocyst, which gets implanted. So zygote divides to form dash, which is implanted in the uterus. So it forms morula, but what gets implanted is the blastocyst. So it is blastocyst, which gets implanted in the uterus. Next one, the structure which provides vascular connection between fetus and uterus. What is the connection between the maternal tissue and the baby or the fetus? Is placenta. So these were the fill in the blank type of questions, that is part of question number 1. Now let us take second question. Draw a well labeled diagram of male reproductive system. Go back to that section where we studied the complete male reproductive system, but quickly if we have to just go over it. The 2 testes, they are present in the scrotal sacs. From the testes, the sperms are poured into a tube, highly coiled tube, epididymis. Epididymis continues through vas deferens, and this vas deferens, it ascends into the abdominal cavity. Here is the urinary bladder, and from the bladder arises urethra. This vas deferens, it takes a turn and it joins this urethra, but before it joins, there is a gland which opens here, the seminal vesicle. Here we made the ampulla region also. So our diagram has to be like the complete detailed diagram, and it connects to the urethra. So there are 2 vas deferens, one from each side is opening into the urethra. Now this urethra, it passes through a muscular structure which is known as the penis, which is the external copulatory organ, and this connection which is here is covered by a large chestnut type gland which is known as prostate gland. Plus at the base of penis there are 2 small glands here which are called Cowper's glands, and then label all these structures: testis, epididymis, this blue gland which we have drawn here is epididymis, this tube which is going up is vasa deferentia, this gland is seminal vesicle, this structure is the bladder, urinary bladder, this big gland is prostate gland, this external structure is penis, the skin of penis at the tip is loose, it is called prepuce, and the entire tip which is very sensitive is known as glans penis. These small glands which are here, they are called Cowper's glands or bulbourethral glands. Cowper's glands. So complete detailed structure of male reproductive system. The third question is, draw a labeled diagram of female reproductive system. Question number 3. female reproductive system. In female reproductive system, same, a pair of ovaries in the abdominal region. From the ovaries, the egg is collected by the fallopian tubes. So, from two sides the fallopian tubes. Each fallopian tube has a funnel-like opening known as infundibulum, and there are finger-like structures at this opening. The base is known as ampulla and isthmus. It opens into a muscular structure called the uterus or womb, and the lower part of the uterus has a narrower tube-like structure which is called cervix. The endometrial lining is folded this part is known as the cervix. This is uterus. And when we make the diagram, we have to label all the parts. The upper part is called the fundus. This middle part of the uterus is the body part, and this narrow part is the cervix. And this gland is ovary, and this is the fallopian tube. The cervix opens into another tube-like structure which is vagina. The opening of vagina, we can show hymen, which may get ruptured later on, and the opening, that is the genital opening of, that is the vaginal opening. Vaginal opening. The opening which is visible in the external genitalia in the vulva part. So, this is the structure with all labels, the layers of uterus, the middle part myometrium, inner part endometrium, and all those labels. Fourth question is, write two major functions each of testis and ovaries. This is question number 4. Functions of ovaries and testes. And only two major functions are asked. One is gamete formation, egg production by the ovaries, and sperm formation or sperm production by the testes, gamete formation. The second function is the sex hormones. So, hormones, female sex hormones, that is estrogen and progesterone. Whereas in case of testes, the male sex hormone, that is testosterone. So, main functions are two, gamete formation and the sex hormone formation. Now, let us take question number 5. Describe the structure of a seminiferous tubule. Now, when structure of seminiferous tubule is asked, obviously we have to draw the section of the seminiferous tubule. So, now when the seminiferous tubules are cut, we see many seminiferous tubules, each lined by the germinal epithelium. There are two types of cells which we see here, cubical cells or cuboidal cells, and some tall pyramid-like cells. All other cells which are cubical or cuboidal, they will undergo spermatogenesis, and these bigger cells, they are called Sertoli cells, which are actually the nurse cells. They provide nourishment to the developing embryo. But we cannot leave it like this. We will have to show some stages. So, we have done spermatogenesis also. These cells divide to form the sperm mother cells, that is spermatogonia, then primary spermatocytes, then secondary spermatocytes. So, here we can keep showing those cells which are here. Now, when the sperms are formed, that is by the process of spermiogenesis, then these sperms, they get embedded in the Sertoli cells. So, the sperms, they with their head, they are embedded so that they can get nourishment from it. So, we will have to show all the stages and we can label it, like we can call it spermatogonium, then primary spermatocyte, then spermatid, and these with tail are the sperms. The diagram is given in our NCERT, which we have to observe that all the stages should be visible in the diagram. And whenever we draw this, though the question is related to seminiferous tubule, never miss drawing these cells, which are Leydig's cells or interstitial cells. These are the cells which produce testosterone. So, things inside the seminiferous tubule as well as the Leydig cells. This was question number 5. Sixth, what is spermatogenesis? Briefly describe the process. We have done the complete process of spermatogenesis, that is germinal epithelium here, 3 phases of the first part, or before that let us write down what are the stages. First part is formation of spermatid and second is spermiogenesis. This is completed in 3 steps or 3 phases, that is multiplication phase, then growth phase and maturation. So, before we draw, we need to know which all stages we have to describe. Now, these germinal cells or germinal epithelial cells, they undergo multiplication, and this multiplication takes place by mitotic division only. So, these cells are diploid, these cells are also diploid, they again undergo mitosis. These cells which we get, they are the mother cells. They are called spermatogonia or sperm mother cells. Half of them act as type A cells, half of them act as type B cells. A cells are the stem cells, they will only undergo multiplication, and B type, they will participate in the process. Now this, it enters the next phase, that is growth phase. Growth, as the name suggests, there is accumulation of proteins for which RNA synthesis, so increase in size, but the increase in size is not significant. In case of oogenesis, we saw the size increases many folds because of yolk accumulation. In this case, there is no yolk which is formed, and it is simply grown in size because of protein accumulation. This is known as the primary spermatocyte. This is also diploid. Now it enters the third phase, that is maturation phase. Maturation phase is meiosis. So it undergoes meiosis 1 first, and we get 2 haploid cells. Meiosis 1 is reductional division. These haploid cells are known as secondary spermatocytes. They are haploid, formed as a result of meiosis 1. Both undergo meiosis 2, and we get haploid, all haploid. These cells are known as spermatids. First part done, that is formation of spermatid by the process of multiplication, growth and maturation. Now spermiogenesis. How would these haploid circular spermatids change into the motile functional sperms? So all these will change into these motile sperms. This process is spermiogenesis, in which we have to write, describe brief process. Okay, so a description is also to be given, not only diagram. So we have to write at every step. So in this spermiogenesis, we have to write down all those steps like nucleus condenses, it becomes long, Golgi body divides into two parts, one part forms the acrosome, other part degenerates, it is known as Golgi rest, mitochondria becomes ribbon like, form the nebuncon, centriole, two proximal just beneath the nucleus, and distal which gives rise to axial filaments. So head part, neck part, middle piece and end, all these are formed by the process of spermiogenesis. So all those changes also need to be mentioned. What is spermatogenesis? The process of sperm formation. Briefly describe the process. So process means all the steps, changes in spermiogenesis and this diagram. This will complete our answer. Let us take the next question now, that is question number 7. Seventh question is, name the hormones involved in regulation of spermatogenesis. Hormones for spermatogenesis. The first hormone is gonadotropin releasing factor or releasing hormone. It stimulates the pituitary to start secreting FSH. So next hormone is FSH. Let us keep talking about their functions also. What is the role of gonadotropin releasing factor? It stimulates pituitary to secrete FSH. So now FSH is produced. What is the job of FSH? It stimulates germinal epithelium to do what? To start division, to start multiplication, and multiplication is the first step of spermatogenesis. So it initiates spermatogenesis. One more hormone, ICSH, interstitial cell stimulating hormone, again from pituitary. It stimulates Leydig's cells to do what? To secrete testosterone. So now testosterone is also added, and testosterone's role is to increase the rate of spermatogenesis. And this starts at puberty. So gonadotropin releasing factor stimulates pituitary to release FSH. FSH stimulates the germinal epithelium of seminiferous tubule to start multiplication. That means spermatogenesis begins. ICSH from pituitary, that is interstitial cell stimulating hormone, stimulates Leydig cells, and these Leydig cells produce testosterone, and testosterone increase the rate of spermatogenesis. So these hormones regulate the complete process of spermatogenesis. So this was our question number 7. Now 8th question. Question 8 says, define spermiogenesis and spermiation. Spermiogenesis and spermiation. Spermiogenesis is transformation of haploid circular spermatids into functional motile sperms. So these spermatids, they are circular, though they are haploid, but they don't even look like sperms. So this process is conversion of those haploid circular cells into sperms, functional motile sperms. This is spermiogenesis. Spermiation. Spermiation is release of sperms, fully formed sperms from Sertoli cells. Release of fully formed sperms from Sertoli cells. Just now we talked about that after this spermatogenesis and all, these sperms, they get embedded in this Sertoli cells till they are fully formed. Once they are fully formed, they detach from the Sertoli cells, and once they detach, they come into this, suppose this is the complete seminiferous tubule, they will come into the lumen, and now they will be conducted from seminiferous tubule through rete testis, vasa efferentia into the epididymis. So once they are fully formed, they will come here. So the process of release of sperm from Sertoli cells is known as spermiation. Ninth question. Draw a labeled diagram of sperm. Again, we have discussed the detailed diagram of sperm. There are three parts. The upper part has acrosome and nucleus. This part is known as the head part. There is acrosome here, which is made up of or made from Golgi body. This part has the condensed elongated nucleus, and the nucleus has a depression here. This part is the head part. Then there is a slight depression, which is known as the neck. In this depression, there are two things. One centriole is here in the depression of the nucleus, which is the proximal centriole, and in this neck region are the distal centriole, from which arise the axial filaments. So this is acrosome, this is nucleus. The first or the upper centriole is known as proximal centriole. These lower ones, which are in the neck region, distal centriole, and these fibers which arise from the distal centriole are axial filaments. Plasma membrane is going to continue in this part. Around the axial filaments are the spiral mitochondria, which are known as Nebenкern. This is Nebenкern. They are surrounding this axial filaments, and the remaining part is the tail part. There are two more membranes. The complete acrosome and upper half of the nucleus, it is protected by an additional membrane here. This membrane is known as galea, and one more membrane is there, which is lower, which covers the lower half of the nucleus and the complete middle piece. This membrane is known as manchete, which is covering the lower half of the nucleus and middle piece. So now this part which has Nebenкern is the middle piece, and the part which is after that is the tail region. So, all these structures must be there in the diagram of the human sperm. 10th question.